Ever try to tell two bacteria apart? Ugh, talk about a challenge, right? It's way harder than it looks. But you know what? Scientists, they've got these, really clever tricks to tell them apart. And today, we're going to dive into one of them, the Bile Esculin Test.
Yeah, think of it kind of like a detective's tool, but for the microscopic world.
Exactly. And get this, we've got these research excerpts, and we're going to break them down for you. So consider us your, research assistants for the day.
We're here to make sure you get the good stuff, the important hows and whys of this test.
Okay, so imagine this. You've got a Petri dish, standard science stuff, right? But wait, there's a twist. Instead of regular jelly, we've got something called Bile Esculin agar. And hold on to your hats because this stuff's got bile salts in it.
Now, these bile salts are pretty interesting. Most bacteria, they can't stand the stuff. It's like a picky eater, but for microbes.
So basically, these bile salts, they act like a filter. They only let certain types of bacteria through, those tough enough to handle them.
Think of it like a bacterial obstacle course.
Love it. And for those bacteria that make it past the bile salts, there's another hurdle waiting. Esculin. This is where things get really cool, because if a bacterium can break down this esculin, it triggers a reaction that, get this, turns the agar black.
Yep, that color change, that's like a big flashing sign telling scientists we've got a positive result.
It's like a bacterial bat signal.
Exactly, and it tells us that this particular bacteria can handle bile, and it can break down esculin. That narrows things down quite a bit.
So for example, let's say we're trying to see if we've got Enterococci hanging around. That's a type of bacteria that can be found in our gut.
Some strains of which, by the way, can be pretty nasty if they end up in the wrong place.
Yeah, no fun at all.
Yeah.
Especially in hospitals, where they can cause some serious infections. Being able to spot them quickly, that's a game changer when it comes to treatment.
And that's one of the things that makes this test, which seems pretty simple on the surface, so important. It's not just about identifying bacteria in the lab, it's about understanding what's happening in the real world. Hospitals, our food, even inside of us.
It's like having a decoder ring for the microbial world.
Yes.
But how does this color-changing magic actually work?
Good question. Let's get into the chemistry behind it all. It's actually really fascinating.
So this color-changing thing, it's like the key to the whole shebang.
Right.
But how does it actually work? What's the science behind it?
Well, remember esculin, the stuff some bacteria can break down. When that happens, it makes this other thing called esculetin.
Esculetin, right, got it. So what's up with esculetin? What's it do?
It's just esculetin's just hanging out in the agar, kind of by itself. But then we add in this other ingredient, ferric citrate. And ferric citrate, well, that's where the party starts. It loves to react, always looking for a compound to pair up with. And guess what? It just so happens to have a thing for esculetin.
A match made in Petri dish heaven.
Right. And when these two lovebirds get together, they react. And that new compound they make, that's our signal. That's what gives the agar that distinctive black color.
It's a love story. The bacteria break down esculin, which makes esculetin, and then ferric citrate swoops in and bam! Black precipitate. Case closed.
Exactly. And even a little bit of bacterial activity, even a tiny bit, can make that color change happen. That's why this test is so good at sniffing these microbes out.
It's like a microscopic alarm system going off if those bacteria are up to something sneaky.
Right.
So, okay, let's say we're back in the lab, all set up, Bile Esculin agar ready to go. What happens if we introduce, let's say, Streptococcus pyogenes? The strep throat culprit.
Streptococcus pyogenes. Tough guy in the throat, but a bit of a lightweight when it comes to this agar. You see, it can't handle those bile salts.
Like trying to get past a bouncer with no ID. It's not happening.
Exactly. No black precipitate, no party for Streptococcus pyogenes. Negative result. But now, let's try a different character. Enterococcus faecalis usually hangs out in our gut.
That's the one that can cause trouble if it gets out of line, right?
That's the one. But this guy, Enterococcus faecalis, he's got the right stuff. Handles the bile salts, no sweat. Waltzes right past them and heads straight for the esculin.
And because it breaks down that esculin, we get our chain reaction, the agar turns black, and there's our answer.
Positive result.
Oh.
And just like that, with one test, we've started to sort out the bacteria, even though they might look pretty similar under a microscope.
It's pretty wild how much we can learn from just one test, huh?
Yeah, it really is something else.
It's basically a bacterial ID check, right?
In a way, I guess you could say that, but we do have to remember this test, it's just a starting point.
Okay, so it's not giving us the whole picture.
Exactly. This is more like narrowing down the suspects, right?
So we know it's a bird, but is it a robin or a hawk? Something like that.
Yeah, that's a good way to put it. We need more tests to really nail it down.
Makes sense. So Even though it's got its limits, this Bile Esculin test, it really shows how creative scientists can be when they're trying to figure out this hidden world of microbes.
It really does.
And it gets you thinking, could we use this kind of approach in other areas too? I mean, we're basically figuring something out based on whether it can survive in a certain place and do a certain thing right.
Now, that's an interesting thought.
Right.
It does make you wonder about the possibilities beyond just bacteria.
Yeah.
What if we could use this kind of selective approach for, like, environmental stuff?
Oh, I see what you mean. Like using it to find organisms that can clean up pollution.
Exactly. Or maybe even in materials science, finding microorganisms that could help break down certain plastics.
That's amazing. So something that started with identifying bacteria could have all these other uses.
It just shows you sometimes the biggest discoveries, they come from the most unexpected places. The more we learn about this tiny world of microbes, the more we can do.
It's like a whole universe just waiting to be discovered right there in a petri dish.
It really is.
And who knows what other mysteries are hiding in there.
Well, that's what keeps us scientists coming back for more, isn't it? Always more to learn.
I love that. Well said. And on that note, I want to thank you all for joining us on this deep dive into the Bile Esculin test.
It's been a pleasure.
Until next time.
Yeah.
Keep those brains buzzing and stay curious.